Hello, my name is Katie Mazalkova.
And my name is William Crugnola, and we are here today to talk about aortic-derived mesenchymal stem cells, a novel target for drug-mediated atherosclerosis cessation.
In the time it takes us to give you this presentation, nearly 400 people will have died from some form of cardiovascular disease. Cardiovascular disease is currently the leading cause of death worldwide, and according to Amarenko 2004 and Gui 2012, the primary cause of cardiovascular disease is atherosclerosis. Atherosclerosis is prevalent today due to the lack of an effective treatment option, which can be attributed to the lack of understanding surrounding its complex molecular mechanisms. A crucial initiating factor of atherosclerosis is the creation of foam cells mediated by scavenger receptors. The obvious solution to block these receptors is impossible without threatening their natural homeostatic roles. We must therefore find an alternative method in order to create a superior treatment.
Atherosclerosis isn't simply the accumulation and hardening of fatty plaque in the arteries. It's a complex cascade of events that contributes to progressive and deadly disease. Atherosclerosis is a multi-step process that is initiated through the formation of vascular lesions, which facilitate the infiltration of low-density lipoprotein, otherwise known as LDL, into the intima through endothelial leaky junctions. When oxidized in the intima, LDL not only promotes the adhesion of additional LDL to the endothelium, it also promotes the migration of macrophages to the intima. The perpetual migration of macrophages and LDL to the intima contributes to the ever-growing vascular lesion. Vascular remodeling initially attempts to withhold the growth of the lesion towards the lumen, which is a process that obstructs blood flow. However, the growth of the lesion eventually reaches a point where vascular remodeling can no longer withhold its growth, at which point the lesion protrudes outwards into the lumen and obstructs laminar blood flow. Blood flow over the thickened vessel wall induces abnormal shear stress, which activates intimal mechanosensors to upregulate reactive oxygen species, or ROS, which in turn oxidizes LDL. Oxidized LDL can activate local macrophage scavenger receptors, SRA1 or CD36, which induce macrophage phagocytosis of oxidized LDL, causing macrophages to transform into foam cells.
Additionally, SRA1 causes the polarization from the anti-inflammatory M2 phenotype to the pro-inflammatory M1 phenotype. M1 macrophages are characterized by the production of pro-inflammatory cytokines, IFN-gamma, IL-12, and TNF-alpha.
Pro-inflammatory cytokines, such as the ones Katie just mentioned, have the ability to activate the M1, are characterized by the M1 phenotype. The initial activation of the M1 phenotype by oxidized LDL induces a positive feedback system in which these pro-inflammatory cytokines produced by the M1 phenotype polarize nearby M2 macrophages back to the M1 phenotype and cause the production of additional pro-inflammatory cytokines.
Adhesion molecules ICAM1 and VCAM1 are critical for T cell activation and for leukocyte recruitment, showing their important role in an effective immune response. They have also been shown to recruit mesenchymal stem cells to the heart by adhesion to the endothelium, thereby providing a role in cardiac tissue repair. However, in vascular lesions, these molecules cause the adherence of newly created foam cells, further inhibiting their migration and contributing to atherosclerosis. As previously stated, the key mechanism in this process that is still misunderstood is foam cell creation. This contributes to the ever-growing vascular lesion and will inevitably cause heart attack and stroke in millions.
The significant death rate associated with atherosclerosis is indicative that current treatment methods are ineffective. All current treatment methods share an important common flaw. None specifically target macrophage phagocytosis of oxidized LDL, which is the primary source of foam cell accumulation in atherosclerosis. But how do we stop macrophage phagocytosis of oxidized LDL? Well, in recent years, bone marrow mesenchymal stem cells have garnered significant attention in the research community for their potent anti-inflammatory characteristics. Because the M1 phenotype is shown to have greater phagocytosis than the M2 phenotype, the ability of bone marrow mesenchymal stem cells to promote the M2 phenotype makes them an attractive target to hinder foam cell creation and atherosclerosis progression.
Mesenchymal stem cells derived from the aorta, known as MAOs, are naturally present at the site of atherosclerosis formation. We hypothesize that, like D1 mesenchymal stem cells, MAOs also have an anti-inflammatory role mediated by adhesion molecules. However, due to the significant lack of literature surrounding MAO function, we decided to focus our study on investigating their potential role as treatments for atherosclerosis.
The initial goal of our study was to compare the effect of MAO to D1 on bone marrow macrophage phagocytosis. The second goal was to evaluate the ability of MAO to promote either the pro-inflammatory M1 phenotype or the anti-inflammatory M2 phenotype in macrophages. The third goal was to use a gene screening to measure the expression of a wide array of adhesion molecules after MAO interaction with BMMO medium. And the final goal was to observe patterns in adhesion molecule gene expression from the gene screening in order to identify a possible mediator of the MAO-BMMO interaction.
We used zymosan bioparticles as markers in a macrophage phagocytosis assay. We ran standard ELISA and Grice tests to measure the concentrations of known M1 markers, nitric oxide, and TNF-alpha. We also used a PCR plate to detect the expression of adhesion molecules as a result of the MAO-BMMO interaction. All our cell lines were direct cultures from HCCC, and we used one-way ANOVAs and GraphPad PRISM-6 for our statistical analyses.
Here are the results of the phagocytosis assay. We categorized the magnitude of macrophage phagocytosis into 4 distinct categories based on the association of these fluorescent green zymosan bioparticles with fluorescent blue macrophage nuclei. Each of the 4 groups was assigned as color, which are represented in the pie chart shown here. In the D1 BMMO co-culture, shown here and here, D1 had a negligible effect on macrophage phagocytosis, which affirms the expected results from the positive control. In the MAO-BMMO co-culture shown here and here, MAO significantly increased macrophage phagocytosis, which, in the presence of oxidized LDL, would promote foam cell creation and atherosclerosis progression. These results also indicate MAO actually has a pro-inflammatory role. These results came as a shock to us, as they were contrary to our original assumption that MAO, like D1, had an anti-inflammatory role. As a result of these findings, we decided to refocus our project towards investigating the pro-inflammatory functions of MAO.
These are the results of an ELISA used to measure the effect of D1 and MAOs on production of TNF-alpha by the macrophages. As expected, D1 mesenchymal stem cells suppressed the macrophage's ability to produce TNF-alpha. However, the MAO showed increased TNF-alpha concentrations, showing that they may actually promote the M1 phenotype. In order to ensure the validity of our results, we also tested another known M1 marker, nitric oxide.
To measure the concentration of nitric oxide, we used a Grice reaction test. Shown here, D1 slightly reduced nitric oxide production by bone marrow macrophages, again affirming the expected results from D1. Both MAO and BMMO alone had relatively low production of nitric oxide. However, the combination of the two, shown here, resulted in a significantly higher nitric oxide output, indicating the MAO-BMMO interaction not only promotes nitric oxide production, but the M1 phenotype as well. After using the Grice and ELISA to confirm that MAO promotes the M1 phenotype, the next logical step was to investigate the mechanism by which MAO promotes the M1 phenotype.
This scatter plot shows the expression of adhesion molecules as a result of the MAO activation of BMMO. The blue dots indicate the downregulated genes, and the red dots indicate the genes that were upregulated. 3 specific molecules, P-selectin, E-selectin, and CCL5, were immensely upregulated, showing that they may be potential mediators of the MAO-BMMO interaction.
Here's a table indicating the numerical expression of adhesion molecules from the gene screening that was represented in the scatter plot in the previous slide. Adhesion molecules were separated into 2 main subcategories based on their general function: cell-to-cell adhesion and leukocyte recruitment. Both cell-to-cell adhesion and leukocyte recruitment molecules were shown to be downregulated across the board, indicating that neither type is inherently upregulated by the MAO-BMMO interaction. However, if we look specifically at E-selectin, P-selectin, and CCL5, we saw that all 3 were significantly upregulated, while adhesion molecules that serve similar functions were downregulated. These results suggest an important and specific role for all 3 adhesion molecules in the MAO-BMMO interaction.
There are several important takeaways from our study. First, we identified that MAO promote the M1 phenotype and macrophage phagocytosis, which is contrary to a common belief that all mesenchymal stem cells have an anti-inflammatory role. We also identified P-selectin, E-selectin, and CCL5 as potential mediators of the MAO-BMMO interaction. And we identified the respective receptors of these molecules as novel targets for drug-mediated hindrance of foam cell creation and of atherosclerosis progression. The knockout of these genes would verify their mechanistic importance and would bring us closer to understanding the science behind a superior treatment.
Lastly, here where the results of our study indicate MAO fits into atherosclerosis progression. We showed MAO promotes the pro-inflammatory M1 phenotype, which constitutes the production of pro-inflammatory cytokines. The activation of adhesion molecules by these pro-inflammatory cytokines inhibits the migration of foam cells, the creation of which is also promoted by MAO through their acceleration of macrophage phagocytosis of oxidized LDL.
These two functions of MAO elucidated by this study enhance our understanding of the mechanisms which mediate atherosclerosis progression. This new information can bring science and society closer to an effective new treatment for the millions of people suffering from cardiovascular disease, both today and for years to come. We would especially like to thank the George Washington University, the Siemens Foundation, and Discovery Education.
We'd also like to thank our scientific mentor, our science research teachers, and of course our parents.
Thank you.
Thank you.